So I went through this text on Friday, and it's a lot easier to understand in the diagram, but refer back to the text here to fill in this diagram. So we're talking about sigma factor regulation, and remember what sigma factors are. They're the proteins that orient the RNA polymerase onto the DNA in order to get transcription. And it's another level of regulation. So we've been talking about activator and repressor proteins, which are different from sigma factors. And we've talked about sigma factors, and we're bringing them back up again because it's another type of control. So these alternate sigma factors. And the example that we're talking about is in a heat shock response, which really represents a lot of stress. These heat shock proteins were first identified when bacteria and our cells exposed to heat, we produce these heat shock proteins. But a lot of stress produce these heat shock proteins. And a lot of them are involved with chaperone repair of proteins. So we've talked about both of those in the days past. Okay, so on the left side of the diagram, kind of in bluish, is kind of the control condition under low heat. And then on the right side of the diagram is what happens to the cells when you expose them to high heat. So 30 degrees Celsius, a little bit warmer than room temperature. 42 degrees Celsius is over 100 degrees Fahrenheit, pretty darn hot. Okay, so in bacteria, I say under low heat, the text says messenger RNA secondary structure. Okay, so here is your messenger RNA that is not just a single strand. Typically, it is a single strand when it is being transcribed. When it forms into secondary structure, it interrupts translation from messenger RNA into the protein. Okay. Nevertheless, let's see. Hold on just a second. The control is at the translational level. So there's not much of this protein produced because of this secondary protein. And so that's one level of control. And this is the sigma factor. This is the factor that is eventually going to help, here's the RNA polymerase, dock onto the promoter of these heat shock genes. Okay? So the green is the is the sigma factor. And when it's produced at low temperatures, 30 degrees Celsius, these chaperone proteins immediately dock onto it and degrade it. And why do the chaperone proteins do such a thing to a sigma factor? There's nothing else for them to do. There's no damaged proteins in the area because of the low temperature. Okay? So those are kind of two effects. One, you get the secondary structure, so you get little production of the sigma factor. And then the second reason why there's not much of the sigma factor at low temperature at 30 degrees Celsius is these chaperone proteins, which are sitting around with everything's great, no damaged proteins, so I'm going to dock to this chaperone, to this sigma factor, and degrade it. That's a little bit me talking down to it. That's not quite the anthropomorphic is the word I'm talking about. But it is part of it. There's no damaged proteins, and so these chaperone proteins degrade the sigma factors. Okay, under high heat conditions, so over here your text says secondary structure is now gone, and so you get a lot of sigma factor production, translation into protein. And now it's 42 degrees. There is a lot of damaged proteins in the cell, a lot of denatured proteins. Now the chaperone proteins are busy doing their normal thing, and that is docking to these damaged proteins and attempting to refold those proteins, like we talked about last week. Okay, so now there's two reasons why you get a lot of sigma production. And that is production without secondary structure, and now it's not being degraded by the chaperone proteins. So now you have the sigma factor orienting the RNA polymerase to those particular promoters that produce more chaperone proteins, DNA K, GRPE, DNA J, and you get this massive upregulation of stress genes in response to heat. And so it's a kind of a nice example of, we're talking about regulation of gene expression in this chapter. Well, here's regulation at the level of translation. So it's regulating at the level of messenger RNA. And it's just kind of another example or another opportunity to see sigma factors in the role of regulating whether that whether that RNA polymerase is going to dock to that promoter. And then just another opportunity to look at the role of chaperone proteins, these heat shock proteins. So, there is your text here describing that, and here is your figure. And so, any questions on that? Okay. So, in the last 10 years or so, a number of new types of RNA have been discovered that were not unknown, part of the non-coding DNA that's just transcribed to messenger RNA, transcribed to RNA that's not messenger, transfer, or ribosomal RNA. What are these RNAs? And so small regulatory RNAs is one pretty broad category. Regulate the transcription and stability of messenger RNAs. And so here's another level of regulation. So the advantage of using RNA instead of protein in regulation doesn't require protein synthesis. You don't have to synthesize that protein and then regulate. Your RNA is already is just one step faster and regulating at the RNA level. And a lot of them have to do with them being the antisense of the gene that they're regulating. So when you say antisense, that means that they are going to base pair match with the sense strand being the messenger RNA that is going to be expressed into protein. So these regulatory RNAs have the antisense and will bind, and there is their regulation. If they bind to messenger RNA, that messenger RNA is not going to be translated. So sometimes it stabilizes the target messenger RNA, or it makes it susceptible to degradation. So in some cases, it actually helps the expression of the messenger RNA. Okay, so the example here is Staph aureus. Do you have that little text at the bottom in your outline? Okay, good. Staph aureus infects human. It's a human pathogen. And it's interesting, it actually, during some point in the infection, in the latter stages of the infection, Staph aureus shuts down its virulence gene production. It no longer wants to express those toxin genes. And so one of the example of those virulence genes are the genes that code for surface attachment. And so here's the example of how small RNAs shut down virulence gene production in the latter stages of an infection. So it's already wiped out, it's already carried out its infection, multiplied the organism by a factor of 10 to the 10th, and now it's ready to move on, either to another part of the body or to another target, another patient. Okay, so I just put this note here that whatever error there was, and it was in the color of the second edition of this illustration, is now corrected in the third edition. So if anybody's got the second edition, you can see me about, there's a black and red problem in the figure. Okay, so here is your regulatory small RNA, and it is it's in secondary structure. So it's base pair matching with itself and forming a hairpin loop structure, which is commonly formed in RNA molecules. And then here is the gene, the messenger RNA, that needs to be translated. Here's the Ch10 sequence, which is part of the ribosome recognizing where it should translate. And I've got some text in here. So what it does, this regulatory RNA, actually, if you'll look at, this is in light blue, the messenger RNA that needs to be translated, and the regulatory RNA is in dark blue. This thing unfolds and base pair matches. It is the antisense sequence of the sense sequence, and therefore will base pair match. Well, if this is going to be now double-stranded, your messenger RNA cannot be translated. Your ribosome can't dock onto the messenger RNA because now you've got double-stranded RNA. It's not its template. And so I say it crumples the stem loop of the target. Well, this had been in the stem loop structure. Now it's gone. And now you've got this double-stranded, no access. So the double strand prevents the ribosome from binding. So you have the X right there. So there you go. It stopped translation. And this is the way Staph aureus shuts down. It no longer wants to attach to another cell. It wants to get out of the body. And then thirdly, this double-stranded structure now becomes template, becomes substrate, becomes target, different ways of saying the same thing, for RNase 3. RNase 3 is an RNA digesting enzyme, and so it's going to digest that messenger RNA. So this entire messenger RNA is now gone and then degraded into nucleotides. And so this is a way these small regulatory RNA molecules, what's in dark blue, can regulate at the level of translation. And so we're kind of building up a whole series of ways that bacteria can, very elegantly and sophisticated, having these multiple layers of being able to regulate, whether the DNA is transcribed, now whether the RNA is translated. And using multiple chemicals, biochemicals to do it. Now we're looking at single-stranded small RNA, small regulatory RNA molecules that can carry out this regulation. End result, messenger RNA is degraded, i.e. the virulence genes are not expressed. Those surface attachment genes don't need to attach to the surface of the cell anymore. Okay, so we're going to put a few of these things together and look at chemotaxis again. And we've talked a lot about a lot of this information already, so it's kind of integrating a few things together. When I say integrated, these are integrated circuits, so multiple operons being controlled by the same regulatory protein, for example, the regulon that we've talked about. So gene switches can regulate, quote, decision-making processes. So remember, chemotaxis is where modal bacteria can swim towards chemical attractants or away from chemical toxins. So chemorepellents, repellents are attractants. Chemical attractants, chemical repellents. And I'd like you to remember that in that structure of that incredibly sophisticated biological motor, there's a switch. And the switch turns it from spinning counterclockwise to spinning clockwise. And the fact that there is a protein switch is what I'm bringing up. And this protein switch is controlled by a regulatory protein called chemotaxis Y protein. So, bringing in what you already know about if that flagellar motor is turning clockwise, it is causing the bacteria to tumble. and if it is turning counterclockwise, it runs. It's the chemotaxis Y protein that controls that switch. So when chemotaxis Y is phosphorylated, it switches the motor to clockwise. When it's dephosphorylated, it switches to counterclockwise. So the simplicity of the system is marvelous. It's just the presence and absence of a phosphate group on this protein that then affects that switch protein in the rotor. Okay, there's this next level of sophistication, and that is there are these methyl-accepting chemotaxis proteins which are clustered at the cell poles, which actually binds those chemicals, certainly binds the attractants. And so now we're looking at not phosphorylation, but another level of modulation of the system, and that is methylation and demethylation of these methyl-accepting chemotaxis proteins. Kind of a mouthful. And so this actually gives you another level of control, not just on-off, but modulating your on-off. And we'll explain that in a second. And matter of fact, right now in text form, and then a little bit later in a diagram form. So once the cell encounters an attractant and starts swimming, it must encounter higher concentrations of that attractant to keep swimming. Okay? And so it's not just on-off, it is sensing concentration differences of that attractant. And it will only continue swimming if it is only swimming in the direction of the higher concentration. So pretty neat, pretty sophisticated ability to sense concentration differences. And in the text, it gives us one of my favorite illustrations, and that is after work, you get out of your car, it's 5:30, you're starving, you have nothing for dinner, but you smell one of your neighbors having a barbecue somewhere. And you are going to go find this barbecue and invite yourself over because that's your only option. So you kind of walk in this direction and no, it's getting less. The concentration of the barbecue smell is less. So you find, and you can do this, right? It's stronger and stronger smell. So think of yourself as a bacterium, and that is what's happening. He is going to a barbecue. That bacterium is inviting himself over to the barbecue, just like you will at 5:30 when you have no other options. So read that over in the text. See if that helps you connect with this behavior. It did me. Okay. Here is the diagram, and this is an improved diagram from the second division. It makes it a little bit more clear, but so is, I have to admit, my simplification. I will give you kind of a brief overview and say there is a cascade of events. Here is this protein. Remember, this is a sensory protein that is sensing information on the outside of the cell and transmitting it into the cytoplasm. And so typically it is a transmembrane protein. And it initiates a cascade of events through chemotaxis W, chemotaxis A, and the interaction of chemotaxis Z and chemotaxis Y. Okay? So all I want you to do is appreciate that there is a cascade of events that eventually simply phosphorylates chemotaxis Y. So I am focusing on whether chemotaxis Y is phosphorylated or not, which turns on and off that switch of the flagellar motor. Okay, so I say steps below are a slight simplification, and I'm going to ignore the intermediate action of chemotaxis A, chemotaxis Z, and chemotaxis W, but just understand that there's this cascade of events. And realize that it is this methyl carrier protein that is sensing the external chemicals and transmitting the information inside. Okay, so to start off with, we have in the upper left-hand corner the situation where there is no attractant, and so chemotaxis Y is phosphorylated, and so here is the phosphorylated chemotaxis Y, which interacts directly with the switch protein on the flagellar motor, which you can kind of go back and look in that chapter and see there is a component, one of the, how many proteins? I don't remember. Were there 40 different proteins that are interacting in this rotor protein? Interacts with that rotor protein and switches it and causes it to go clockwise and tumble. So that's in the absence of an attractant. So number 2 down here, in the presence of an attractant, and this is the red hot dog from the barbecue that is now bound to the methyl carrier protein, the environmental sensory protein. That causes this cascade of events that causes the dephosphorylation of chemotaxis Y. And when chemotaxis Y is dephosphorylated, it interacts with the rotor to cause counterclockwise movement and swimming. So understand that basic effect. In the presence of an attractant, chemotaxis Y is dephosphorylated, and you get counterclockwise, and you're going to get more runs than falls. Okay, then here's the next level of information, of our modulation. How does it modulate this? It doesn't just turn it on and off. It's responding to a concentration difference. So if the cell does not encounter a higher concentration of attractant, this methyl accepting protein gets methylated. So here you see a methyl group being added directly to a component of the methyl accepting protein. And with that methylation event, that affects whether that attractant will bind or not. And basically, if it's in the same concentration of when it bound in the first place, it does not bind again. The only way you're going to get, and this is what it says down here, the only way the cell keeps running is if the attractant concentration increases. And if it increases, you will get binding to the methyl accepting protein, dephosphorylation of chemotaxis Y, and continued swimming of the bacteria. But only if that concentration of the attractant increases. And so here you have this modulation of the on-off switch, or clockwise, counterclockwise switch, which then says it's only going to respond to increasing concentrations. And so if the cell happens to swim into an area where it's decreased, it's going to be tripping and falling. And it will only start swimming again when it encounters higher concentrations. So this is just to explain how the cell responds and can sense different concentrations of the attractant. So pretty cool, amazing structure, and perhaps I have two more. So when you methylate the MCP protein, it desensitizes it. So in other words, that same concentration of attractant does not bind. Only higher concentrations of attractant will bind once it's methylated. Okay, and then lastly, if there is no attractant present, the MCP protein is demethylated. Here's the demethylation. And the entire system is reset. And now it's waiting to encounter any kind of attractant. And so just a little bit more detail on what you basically knew 2/3 of the story. And now it adds this other level of the methylation effect, which allows it to respond to increasing or decreasing concentrations of those attractants. How about any questions on that? Okay, study and then bring those concentrations tomorrow, and I should say continue studying. Okay, This is great. I'm flying through this material. It's still herbal tea. There's nothing fueling this this person here in front of you at 8:30. But somehow we muster. Oh, yes, a question. No, you're just scratching it. Exactly. Oh. Okay, this is a pretty cool photograph. Let's see. It's a computer model. I don't know. This is more closely related to a photograph because all of these different proteins are labeled with a fluorescent protein and are the cell is photographed under different wavelengths of light, which then highlights these different proteins. So it really is pretty representative photograph, which shows a cluster of these methyl carrier proteins which are at one pole of the cells. So, you know, the bacteria are directionally motivated, and so they are typically swimming this is basically the direction that it is swimming in, is towards the head and the tail. And the head, the quote head of the bacterium, has a huge concentration of these methyl carrier proteins, or receptors at one end of the cell. And so these are labeled in orange and yellow. And then, not shown very prominently because it just didn't pick up, but there are a number of light blue proteins which are tagged with a light blue fluorescence antibody that represent the flagellar proteins. But then the main thing that they've got that gives you kind of a connection with what we've been talking about is the phosphorylated form of chemotaxis Y protein is labeled in red, and you can see there is a higher concentration of the phosphorylated form towards this end of the cell compared to that end of the cell. And so even the length of the cell is sensing the difference in concentration of the chemoattractant here, and then 1 micron later, or 1 micron distance, it can tell that difference. And so even the length of the cell that's the kind of resolution, I'm trying to say, that the cell has. And so in this example, it's phosphorylated, and so this cell is either about to or in the midst of tumbling and hopefully finding itself in a higher concentration of the chemoattractant. Now, your second edition has another part of that figure which is really cool, and I don't know why I didn't bring it into this slide. So see that, for your information. It's a nice, a little bit of additional information of the system. Okay, so just to reiterate, we're having a study session tomorrow, Foster Hall Room 231 at 5:15, and we want to finish up with something that we've talked about a fair amount of, and that is quorum sensing. So we talked about quorum sensing in gram-positive bacteria in that transformosome, which is uptaking DNA. So don't bother uptaking DNA unless there is a quorum of cousins, and now you've got some DNA worthwhile taking up. So there's this ability that bacteria have to sense local populations. And local populations influence whether you're going to transcribe your genes, at least those batch of genes that are controlled by quorum sensing. So these are genes that are only turned on when the cell reaches a critical threshold high density, discovered in Vibrio fischeri. And so Dr. Castillo and Dr. Nishiguchi, we've got two professors that study this symbiosis between the fluorescent light-producing bacteria and the squid. And basically, those organisms only turn on the light when there is enough of the bacteria to see the light. So just think about 150 cells turning on their little 1 micron lights. Nobody notices. It's like, turning on the lights, but there's only 150 1 micron cells. The environment doesn't see that light. But when you have 1.5 billion of those cells and you turn on the light, everybody notices. So let's not bother turning on the light at low concentrations. Let's only do it when people or squid will notice. And you can really see this with your naked eye when you've got a lot of cells on a plate, which is what's being shown here. Fluorescent blue-green is this kind of fluorochrome that's produced by this Vibrio fischeri. So how does that work? It's about the language of chemistry and chemistry talking. So the induction, the turning on of that quorum sensing gene, requires the accumulation of these chemicals called autoinducers. And this is the relationship between chemical concentration and cell concentration is direct. So as cells grow in their population, so does the concentration of these autoinducers. And so it's this concentration of chemical that indicates concentration of bacteria. Okay, so at certain high extracellular concentrations, the secreted autoinducer re-enters the cell. So we'll look at that in this diagram. And like I've said, the inducer concentration is chemical concentration directly related to cell concentration. So if cell is high enough, quorum is reached, genes are expressed. Okay, and so we'll look at how this inducer, this autoinducer,